Oh, we are live now. So good afternoon, good morning, good evening to everyone. It's really a great pleasure today for me to introduce the speaker of this wonderful series of seminars. So the worldwide neurodevelopmental series organized by Denis. And in particular today is really my great pleasure to introduce Madeline Lancaster. So Madeline, welcome. So Madeline is originally American, and she studied in California, in Los Angeles, biochemistry. And after that, she actually moved to San Diego, still in California, where she joined for a PhD, the laboratory of Joe Gleason, where mainly she focused on trying to understand more about Wnt signaling pathway and the cilia in the context of ciliopathy and Joubert syndrome. So after completing a PhD with wonderful publication and wonderful studies, she actually decided to do an unusual move. So she actually moved east. We always, we do move west to do our postdoc, while actually Madeline, she moved from the US to Europe. And in particular, she moved for a postdoc in the lab of Jürgen Knoblich in Vienna, where she actually did wonderful work. And in particular, in the lab of Jürgen, she developed a very novel model system, also together with other labs in the world, but she was one of the pioneers developing this novel model system. We all know it now. I mean, cerebral organoids are all very well known now. But at that time, the only thing that we knew was to study actually human cells derived from stem cells, mostly in 2D. So progenitors and neurons were mostly cultured and studied in 2-dimensional. While what she did was to try to develop a 3-dimensional model system. And unexpectedly, actually, this 3 model system was not just a bulk of cells, intermingling in a random way, but by looking carefully at this 3D structure, you could actually spot that they were nicely organized. And organized in a way that in certain, despite being in vitro, could resemble some of the steps that they are taking place in the developing human brain. So the first time that we could actually look in 3 dimensions how progenitors sit together and become neurons and migrate, and in particular human cells. So it was definitely a big advance in understanding how our human cells work. So with this wonderful discoveries, she actually published very nice work with Jürgen, but this was also the basis of a new step that was building her own lab. So to do that, she actually moved to Cambridge, where she is since 2015, where she has her own lab, and of course she built on this model to study a variety of questions, I would say, a pretty broad lab in which they study they ask a lot of questions like evolution, development, disease, and it's not just developing new protocols, what she does, but she's actually doing to, for example, model different brain area or different structure. But what I really appreciate of her work personally is the fact that she always does a proof of principle experiment to show how this model system, this improved model system, can also be important for modeling not only development or evolution, but also to model developmental disorder. And this she did already with the first publication in which she was modeling microcephaly, but later on also neuronal migration using, ethanol exposure. Now COVID-19, recently she used the cortex-derived organoids and CSF to model COVID-19. So I think it's really also to show how this model system can be used for modeling developmental disorders or any type of disorders. So not only science focused, I also want to mention that she, of course, with this successful career and this successful model, she had a lot of funding and prizes and awards. It's a long list, so I will not mention all of them. But maybe couple of worth to mention are very prestigious ERC starting grant and also the EMBO Young Investigator Award. And yes, and with that, I will move to what she will talk today about. She mentioned briefly that to me that she will discuss about how the human brain size is regulated, and touching particularly two topics. One is evolution, and the other one is how sex hormones can also regulate brain development. And yes, so I'm very happy that you are here. Thanks for accepting this invitation, and thanks for being here. And we are really looking forward to your talk.
Thank you, Sylvia. That was a really wonderful introduction. Yeah, let me just make sure my slides are up. Yeah, so so I am going to, like Sylvia said, I'm going to mainly focus my talk on evolution and sex. So let's jump right into it. So my lab is interested in, teasing apart what features are sort of make our brains unique and give us our unique cognitive capacity. So we're interested in a variety of different, aspects of that, but in particular focusing on what features, really are human specific and how that can also inform on neurological disorders, which generally affect these kinds of human-specific capabilities that we have. So these are important, issues, not only because they're just interesting, but also because neuroscience is an area of huge unmet medical need. So around 1 in 4 people every year will experience a mental illness, and there's a huge amount of money being spent on other disorders, but given how prevalent mental health conditions and other neurological conditions are, they receive really a fraction of the funding that other things like cancer research receives. And so one of the reasons for that is also probably sort of a chicken and egg issue, because if there's not very high success rate, it continues to receive less funding and less attention, particularly from pharmaceutical companies. And so one of the reasons I think that this area is exhibits such high failure rates is because of what some people have called the clinical trial cliff. And this affects all areas of human biology, but especially neuroscience, where a number of neurological conditions have been cured in animal models. Spinal cord injury, for example. There have been drugs that have essentially enabled mice to walk again, and yet when they reach the clinical trial stage and go into humans, they fail, unfortunately. And this can often this can be because of issues with like toxicity, but also efficacy. And the reason for that is really because of these human-specific features. And if you think about it, our brains are probably one of the organs that's most unique compared with other animals. So, like I say, we're interested in trying to identify what features make our brains unique. And actually, for the most part, there isn't really, one feature that really sets us apart. And the features that are sort of different are not really qualitative, meaning what I mean by that is that there's not like a part of the brain that only humans have. There aren't clear cell types, for example, that only exist in humans. But one thing is clear, and that is that our brain is very large. And so it's not only just large in terms of its absolute size, but especially when you take into account our body size, which is what this encephalization quotient is demonstrating here. So without going into too much detail about what this really means, I'll just summarize it by saying that this value here of about 7.4, it means that our brains are about 7 times larger than they would be expected to be for our body size. And if you compare that to some of our closest living relatives, the other apes, you can see that our brains are really quite large for the body size that we have. And especially when you compare to, for example, mice, which are one of the more common animal models used. And so the brain size, of course, is related to neuron number. And also glial number, of course, all the supportive cells are equally as important. But I'm going to focus on neurons because that's what we're currently focused on in the lab. And so our brains have around 80 to 100 billion neurons, compared with the around 30 billion in gorilla and only 70 million in mice. And it's really difficult, I think, to sort of wrap our brains around what this number means. When you get into the millions and billions, it's really hard to grasp what a big number that is. So for the purposes of illustration, I've just sort of calculated how many neurons would need to be made over the course of 9 months of gestation per hour in order to reach this number. And full disclosure, that's not how neurogenesis occurs. It's not evenly distributed throughout 9 months of gestation. But just for the purposes of illustration, if you do that, you get 12 to 15 million neurons every hour for the entire 9 months in order to reach that 80 to 100 billion. So it's really quite a mind-boggling number. And so we're interested in that human-specific expansion and how specifically in humans the brain expands so much more than in other animals. And so here I'm just showing a couple of coronal sections through adult human and chimp brain, just to kind of illustrate the size difference. So our brains are around 3 times bigger than chimpanzees and gorilla brains. But the size difference is pretty sort of, it's distributed over the surface, basically, of the brain. So what I mean by that is, if you look at the gray matter here, which is the dark region around the outside and contains the cell bodies, the gray matter doesn't seem to be expanded, it doesn't seem to be thickened in humans compared with other apes. And instead, you just see sort of more gray matter distributed over the surface. And so what that's pointing to is an expansion in the tangential domain, so over the surface of the brain, as opposed to in the radial domain, which would lead to thickened gray matter. So, and I'll come back to that later when I talk about evolution. So in order to understand this human brain expansion, we are interested in human brain development, because of course neuron number is going to be determined by neurogenesis and these important cell fate decisions that happen during development that determine how many neurons are going to be made. So I have to just give a little bit of a background on the developmental stages that we're interested in. And so these are sagittal sections of human developing brain. And you can see it starts out as a neural tube. This is an epithelial tissue, and like all epithelia, it has a fluid-filled lumen on the inside, and it's surrounded by epithelial cells that have, apical basal polarity. And different regions of the neural tube will expand at differing rates, with the more rostral portions here expanding and really ballooning out in humans to give rise to this large cerebral cortex, which is so expanded in humans. And so the wall of this developing telencephalon here has a cytoarchitecture that looks something like this, where you have these epithelial cells, which are called neuroepithelial cells, and divide symmetrically initially to expand their numbers and set up the founder stem cell pool. And then those cells switch to a mode of asymmetric division to self-renew, generate one daughter cell that stays as a stem cell, and these are now called radioglial stem cells. And then another daughter cell that will become more differentiated. And so these can be neurons directly, so they can make neurons directly, which migrate out and position themselves in the cortical plate that becomes the gray matter of the brain. Or different sort of intermediate populations like intermediate progenitors or outer or basal radioglia that can also divide and expand and generate neurons. So we're very interested in the cell fate decisions that happen in these various progenitor populations that help determine how many and what types of neurons are being generated. And then these neurons position themselves in the cortical plate in different layers, and that positioning as well as molecular identity helps determine their projections, and that will establish this very well-organized connectome. And so a functioning human brain requires that all of these sort of prerequisite architecture, cytoarchitecture is present in order to set up sort of the blueprint for the brain. So in order to understand those human-specific processes, we want to have a model that that replicates that kind of architecture as closely as possible. And so this is where cerebral organoids came about when I was a postdoc in Jürgen Knoblich's lab. We wanted to try to model these early events as closely as possible. And so I actually have to take you even further back in development. So I showed you the brain as it develops from the neural tube. But let's go back to the very beginning. So at the very beginning of development, we have a blastocyst forming from the fertilized egg. And the inner cell mass of the blastocyst will give rise to the embryo. And it will undergo a series of morphogenetic and cell fate events that help establish the 3 germ layers. And the ectoderm, part of the ectoderm, will generate the neuroectoderm, which is the precursor to the entire brain. And the neuroectoderm will fold up and close to form this neural tube. So in order to get something in vitro, obviously we want to do this in vitro. We can't study human brain development in vivo. We need to try to mimic these events as closely as possible. And so to do that, we take pluripotent stem cells, which are the equivalent of these inner cell mass cells. We then take them off their 2D dish, and we dissociate them essentially and aggregate them to form these embryoid bodies. Now, embryoid bodies are called embryoid bodies because they start to differentiate spontaneously and make sort of different germ layer identities and sort of try to make an embryo. Obviously, they fail, for a variety of reasons. But they do generate different germ layer identities within that embryoid body. And in particular, they generate very nicely neuroectoderm, which tends to form around the outside. And when these tissues then are placed in a droplet of matrigel, which is a protein-rich gel, an extracellular protein-rich gel, then you get a series of tissue reorganization that then leads to the formation of these nice neural tube-like buds. And these neural tube-like buds can then expand and form very large tissues that we call brain or cerebral organoids. And they become so large because we also introduce some agitation in the form of a spinning bioreactor and orbital shaker, and that allows for better nutrient and oxygen supply. Now, since we published this paper in 2013, like Sylvia mentioned, we've been continuing to improve and modify these protocols. And so, for example, one of the sort of tricks we now use in the lab is a little bit of bioengineering to increase the surface area to volume ratio, and that helps to improve the amount of neuroectoderm that is produced, and therefore improve the reliability of generating neural tissues. And we can get really quite nice tissues here. So what you're looking at here is an organoid, obviously, and you can see these kind of outgrowths here. And so we tend to call these buds. These buds are filled with a fluid-filled cavity. And so essentially what we think is going on here is that each one of these is essentially equivalent to a telencephalic vesicle. So whereas normally in a developing brain in vivo, you should really only have one telencephalic vesicle that splits into the 2 hemispheres, these organoids have multiple of these buds, of these telencephalic vesicle-like buds. And as I say, when you section them, you can see that they have these fluid-filled cavities and a very dense cytoarchitecture. Now, each one of these we can sort of look at in isolation, essentially, and compare to the developing telencephalon in vivo. And we can see that they have a very similar kind of architecture. They can get to be very large. You can see, for example, this lobe here is over 1 millimeter in length and reaches essentially a size similar to a mouse embryonic telencephalon. When you zoom in on the wall of this cortical, developing cortical tissue here, you can see a similar sort of cytoarchitecture to that that you see in vivo. We've also been sort of further modifying these and coming up with newer approaches for modeling later events, for example, in development. This is work carried out by Stefano Giandomenico in the lab, where we were able to culture these organoids for much longer term at the air-liquid interface to improve oxygen and nutrient supply. And therefore support very long-term culture. So we're talking about at least a year or more. And you know, the whole 3D organoids that we were culturing up until this point would tend to show quite a lot of cell death if you just sort of kept them like that for a year or more. They'd get very small. You could see them actually shrinking over time, and essentially most of the neurons would be dead by the end of the year. But instead, what Stefano was able to show is that when cultured at the air-liquid interface, we can keep these neurons alive and well for a very long time. We actually don't know how long because we've not kept them long enough, I think. But we can see also this beautiful long-range projections. So you can see these axons that actually bundle together and project over many millimeters and even centimeters. And he was able to show that when you provide a target tissue for these long-range connections, you can actually get innervation from the human organoid into a mouse tissue. This is a mouse spinal cord with muscle attached to it. And when we stimulate the human organoid, we can even see contractions in the mouse tissue, showing that it's a functional connection. And so this work is published, and there's a lot more to it, of course. If you're interested, you can check out the paper. Now, very recently as well, this is work carried out by Laura Pellegrini, and she was able to generate another region of the telencephalon. So this is sort of focused on cortical tissue, but Laura focused on choroid plexus, which is the part of the brain that generates the cerebrospinal fluid. And she was able to modify our protocol and generate very pure choroid plexus organoids that look a lot like the real thing. And they generate these large fluid-filled sacs. And actually, we were able to collect fluid from those and using mass spec compare with in vivo CSF samples and see a high similarity. And also show that these tissues are surrounded by a barrier that is highly selective and shows the same sort of selectivity that you see in the CNS barrier that protects the brain, you know, from toxic compounds and prevents entry of a lot of therapeutics. So this is now a new model for, you know, looking at drug entry into the brain. I want to just quickly, sort of briefly, take a little diversion into COVID, just because I think it's a topic that's on all of our minds at the moment. And talk about, this is work still carried out by Laura recently, looking at SARS-CoV-2 and trying to determine whether there might be some neurotropism for this virus. And the reason we got into this is because there have been described a number of neurological manifestations in COVID-19. And some of these are, you know, may have nothing to do really with the neurons. They may be more likely because of, you know, vascular abnormalities, or stroke and things like that are probably related more to the vasculature. But there are some sort of worrying signs, like, for example, psychosis even in some of these patients, and what's been just sort of called brain fog, and we don't really know where that's coming from. So there's quite a bit of neurological symptoms that patients experience with this condition. So as a first go at this, we decided to look at the RNA-seq data that we have for our brain organoids and look at the different sort of clusters of cell types and see whether the receptors for this virus are present. And basically what we found is that the receptors seem to be only present in choroid plexus cells, so different choroid plexus cells of the organoid. And this actually matches with in vivo data. So this is from the Allen Brain Atlas. You can see that the receptor is highest in choroid plexus. So to get at what cell types in particular are expressing these receptors, we took all of the choroid plexus cell types from our data and then sub-clustered them out. and found sort of some of the clusters we sort of already knew that were there, but we found a new cluster that we hadn't identified previously, which we're calling lipid-producing choroid plexus, or lipoprotein-producing choroid plexus. And that's because these cells express a high abundance of apolipoproteins. And those are actually the cells that also express these receptors. So we also looked at what was co-expressed with the receptor and found other viral receptors, including other coronavirus receptors. And again, these lipoproteins. So that would suggest that the choroid plexus might be susceptible to this virus if it's got the receptors. So to test that, we first tried just putting in a spike protein pseudotyped virus and found that it did, in fact, infect choroid plexus organoids. And this is just showing you the positive control here. That's a virus carrying a VSVG glycoprotein that will allow it to infect sort of any cell type, and a negative control that lacks the envelope glycoprotein. And we also found that the cells that were infected seemed to be expressing the receptor, as you might expect. Now, what was a bit surprising to us was that we didn't see any infection of cortical tissues. So we didn't see any infection of neurons or glia, or the neural progenitors that are present in these organoids using this pseudotyped virus. But we did see infection with our positive control, telling us that the assay is working. So this could be, now this kind of matches our RNA-seq data, but maybe the lack of infection has something to do with the fact that we use a pseudotyped virus. So we decided then to team up with Leo James's group at the LMB and start working with live virus. And here we've infected organoids that have a mixed identity. So these are telencephalic organoids that express a variety of different telencephalic regional identities, including cortex and choroid plexus. And what we found is that the virus only infected the choroid plexus and did not infect neural tissues. We also looked at whether the choroid plexus could be a productive site of viral infection by looking at SARS-CoV-2 mRNA viral copies over time, and found that these were increasing after inoculation, showing that there seems to be replication. And we also found, when we did staining for this apolipoprotein, that the cells of the choroid plexus that seemed to be susceptible seemed to be those that we found in our RNA-seq data that are also expressing the receptor. So, this makes sense. But again, this data already shows we don't think it's infecting cortical tissue. We also tried cortical tissue on its own so that there's no choroid plexus to potentially, act as a sink for the virus. We still didn't see any actual specific staining. And the only time that we could really see any staining at all was if we upped the amount of virus by at least 10 times and also left it on for longer, so at least 2 days. Then we could start to see a couple of neurons in these organoids, but it was very, very few. So we think that this virus doesn't seem to have very much tropism for neurons, at least using this method. But what does it do to the choroid plexus? So we then looked at later stages and found that over time it seemed that the tight junctions between these choroid plexus cells, which, as I mentioned, they act as a very important barrier to protect the brain, that these seem to be breaking down after infection with this virus. And in fact, we could see that they were functionally broken down, so we can see leakage of fluid out of the organoids. And so this is just sort of a model that we've come up with, which is we think that the virus probably, if it goes systemic, if it actually gets into the blood, then it would have probably easy access to the choroid plexus, because these endothelial cells, this endothelium of the choroid plexus is is very leaky because the endothelial cells are fenestrated. So virus can easily leak across and get to the choroid plexus. And if choroid plexus cells in vivo are as susceptible as we're seeing in vitro, then that that is concerning in terms of this barrier that's so important for protecting the brain. And so this has certain implications in terms of the symptoms patients are experiencing. We think maybe not only will virus then get across, but perhaps more concerning than that is actually that potentially cytokines and immune cells that really shouldn't be getting into the brain would suddenly now have access and lead to symptoms of neuroinflammation, which many of these symptoms do indicate a potential neuroinflammation going on. Okay, so now I want to go to what I really wanted to talk about today, which is these intrinsic and extrinsic regulators of brain size and neuron number. And so I'm going to start by talking about our project on sex hormones. So this is work carried out by Eva Kilava, and she's been interested in this because of the clear sex bias of a number of neurological and neurodevelopmental disorders. So in particular, for example, ADHD and autism spectrum disorder show a clear male bias, whereas there are certain other conditions that seem to show more of a female bias, like, for example, eating disorders. And so this suggests that maybe there seems to be a difference in susceptibility between males and females, and suggests that maybe there's something different about their brains that might explain this difference in susceptibility. Now, the thing I should stress is that males and females are highly overlapping in any sorts of measures that we take. So you have to take very large numbers of people to get anything, to show anything significant. But when you do that, the one sort of measure that does seem to be reliably different on a population level is total brain size. So total brain volume, gray matter volume, white matter volume are on a population level, on average, larger in males than in females. And again, I should stress on an individual level, you can't tell just by looking at an MRI necessarily whether it's a male or female brain. But still, this sort of population level effects suggests maybe there's something a bit different about, neuron number or brain size. And so that's also why we got interested in this topic. And interestingly as well, there's been some investigation of differential gene expression between males and females, and in different brain regions. And what this has shown is actually that the stage at which you see the biggest difference between males and females is actually during development and not so much during postnatal or adulthood. So that would suggest that maybe these differences that lead to these differences in susceptibility might come from developmental events. And so that suggests maybe our organoids could be useful for understanding this. So there's basically two possible ways that sex-based brain differences could come about. One is through genetics, because obviously the difference being XX or XY. So this would be a very sort of intrinsic difference between the cells of the brain. And another is hormones. So hormones being made by the developing gonads. And actually, during development in the male embryo, there is a very large surge of testosterone being made by the fetal testes. And this amount of testosterone being made is actually almost as high as the pubertal surge, which is really quite a lot of testosterone being made at that point. So, there could be quite high amounts of testosterone circulating in male embryos that could influence the brain differently than in female embryos. So basically, to sort of come at this from a very kind of almost naive standpoint and just look at all these different variables, Eva generated organoids from male and female cell lines and then treated them with different sex steroid hormones. So she treated with estrogen, testosterone, and DHT. Now, DHT, dihydrotestosterone, is a sort of more potent form of testosterone that can't be converted to estrogen. So both of these are androgens, or male sex hormones. And then she looked at a variety of different readouts. And the timing that she did this as well was, we chose this timing to try to mimic the timing of that fetal surge that I just mentioned. So at this point in these organoids, they have morphology that appears similar to the timing at which the fetal surge begins. And so I'll jump right into what she found. So she found that essentially she didn't see any differences between the male and female cell lines, the untreated male and female cell lines. But what she did see is that when treated with androgens in particular, so the DHT and the testosterone here, she could see an increase in TBR2 cells. And this is quantified here. And this was true in both male and female cell lines. So it seemed that the androgens had a strong effect regardless of chromosomal complement. She also looked at, so she then looked at, where is this increase coming from? And to do that, we did clonal labeling, so basically injecting into these organoids a virus expressing GFP, a very, very low amount, so at clonal dilution. And in this way, you can label individual radioglial stem cells and then look after some time at how big those clones are. And she found that upon treatment with DHT, the potent androgen, she could see an increase in clone size. To see whether that's going through the androgen receptor, which is what we would expect, she also expressed an androgen, a constitutively active androgen receptor in these cells, and found an increase in the number of cells that were expressing this proliferation marker Ki67. So that suggests that androgens increase proliferation of these progenitors. We then wanted to get at, what's leading to this effect. And so to do that, we did RNA-seq on organoids treated with androgens or control, and then looked for differentially expressed genes. And she found a number of differentially expressed genes. When we did GO-term analysis, we found, terms associated with chromatin remodeling, lots of transcription factors and things, and also HDAC activity. You can see these two HDACs here. So we looked more at the HDAC activity by using an HDAC inhibitor, valproic acid. And so when we treat these organoids with valproic acid, we essentially see the opposite phenotype to that with DHT. We see a decrease in these TBR2 cells in the SVZ. But we can rescue that when we put DHT on, suggesting that these are acting in the same pathway. And so this suggests that there's androgens acting through proliferation. And this is very interesting because in autism, actually, chromatin remodeling and epigenetics have been suggested to be important players in susceptibility to autism. And of course, there's the sex susceptibility. So we think that these two things may actually be directly linked. And interestingly, there was a paper showing that the social deficits in a mouse model of autism could be rescued with HDAC inhibition, further suggesting that these are acting together. We also found a number of terms associated with brain size. You can see microcephaly, macrocephaly, head circumference here. And that links back to what I said before about this difference in brain size on a population level. So brain size being related to neuron number. We also looked at neurons and found that upon treatment with androgens, we see an increase in these SATB2 excitatory neurons. And this is quantified here. So basically what we've shown is that it seems that androgens promote proliferation of these progenitors, leading to an increase in basal progenitors and an increase in excitatory neurons. And this effect seems to be specifically affecting excitatory neurons because we don't see an increase in inhibitory neurons. And that's very interesting because it's also been suggested that in autism there may be an imbalance of excitatory and inhibitory neurons. And so potentially the increased susceptibility in males may have to do with slightly tipping the balance towards more excitatory neurons. And then of course, if you add on top of that genetic risk factors or environmental aspects, then you can see how it may tip the balance all the way to autism. Yeah, so now I'm going to switch gears and move to the evolutionary project that I wanted to talk about. So this is, we basically wanted to, like I say, we're very interested in the human-specific differences. So we wanted to do that. We wanted to look at human brain organoids compared with our closest living relatives, the other great apes. And so there's been a number of studies that have also been interested in this and have looked at various aspects of neurogenesis, done mainly looking at single-cell RNA-seq to look at their transcriptomic differences. But we wanted to look more at sort of the tissue architectural differences. So this is work carried out by Silvia Benito in the lab. And what she did is basically, generate organoids from different species. So we have several different human, chimp, and gorilla lines, but these are just some examples. And what we can see is that generally the organoids, while the protocol worked just as well for these other ape cell lines, the organoids were generally a bit smaller in the other apes. So to get it when this size difference first arises, we looked at the very early stages. So this is just after they've been moved to the neural induction media. And you can see that they look the same, basically, between the different species. But we do just start to see a difference at 5 days after neural induction. And you can see these little telencephalic vesicles, the beginnings of these telencephalic vesicles forming. And overall, the organoids seem to be a bit bigger in human, and in particular, the telencephalic vesicles are a little bit bigger. You can see that here much better, actually. So you can see these are a little bit bigger than in the gorilla or the chimp. And they're bigger because they have a larger apical lumen. So we've traced these out and quantified them, and you can see that the lumen is larger in human than in ape. So going back to our model, what might be going on here? Well, we know that there's an important switch that goes on during development from these neuroepithelial cells that are expanding symmetrically to these asymmetrically dividing radioglial stem cells that generate neurons. And so the mode of expansion here is different. At this stage, they're dividing symmetrically and leading to a tangential expansion, so an expansion in this dimension. Whereas at this stage, they're now dividing asymmetrically, and they're generating more cells in the radial dimension, so they're basically thickening the cortical wall. So could the difference between human and ape be because of a difference in timing of this switch? So the way to look at this is to look at these more differentiated cell types, these intermediate progenitors, these TBR2 positive intermediate progenitors, and neurons. The simplest explanation would be that maybe in ape organoids, they're already generating these more differentiated cell types. So they've already made the switch. So to look at that, we stained for TBR2, a marker of the intermediate progenitors, and double cortin, a marker of neurons. And what we found was quite surprising. We did not see these cells already being generated in the ape organoids. They do come on later, just as you would expect, just like in human, but they don't seem to be made at this early time point when the size difference is already apparent. So clearly, an earlier switch is not the explanation. So we then thought about, well, maybe this tissue architecture relates more to the cell shape. And so to look at that, we did sparse labeling with a virus carrying a GFP. And we could see that at this early time point before the size difference appears, that in both cases, both human and ape, these neuroepithelial cells are nicely columnar in shape. And if you stain for ZO1, which stains around the apical surface of these cells, you can see these apical contacts, and you can see that they're quite large. In comparison to at the day 5 time point, when we can see the difference in size, you can see the cell shape is different. And I think it becomes even more apparent when you look at the ZO1. We can really see that in the ape, the apical process has thinned out quite a bit, and these apical contacts are much more narrow. And then later, the human organoids also, these cells also then thin out and become more elongated, and eventually, of course, they switch to generating neurons and intermediate progenitors. So, and this is just quantified here, showing that, so in both cases, you have this change in cell shape over time, but it seems to be delayed in human. So basically, what we're seeing here is that you have initially these neuroepithelial cells dividing symmetrically and expanding. Then you see a change in cell shape in the ape a little bit earlier than in the human. It does happen in the human, but a bit later. And the interesting thing is that this change in cell shape is happening while these cells are still neuroepithelial. So they have not made any of the other changes that come along with becoming radioglia. They have not started expressing glial markers. They haven't changed their identity at all. And they haven't changed their fate. They're still dividing symmetrically. It's really just their shape that's changed here. But that shape change can affect the number of progenitors that are there and lead to a slightly increased number of founder stem cells here in the human, so that once they both do switch to making neurons, in the human, you have slightly more progenitors, and that will then lead to increased neurons. And the interesting thing here as well is that just that this transition state, I guess, the fact that cell shape changes first had not been seen before in mice, for example. So to get at what the molecular mechanism is of this, we did an RNA-seq experiment where we compared human and gorilla organoids at various time points across different batches, different replicates and different time points. And what we found is that we saw quite a lot of overlap early on, but right at day 5, when we start to see this shape change, we also start to see these separating out. They become even more separate here, and then they're very different at the end. And these differences also replicate those seen by others who've done, other RNA-seq analyses and have shown differences in a rate of neurogenesis later on. But this is what we were most interested in because it relates to these differences in cell shape and tissue shape. And so we focused in on these stages here to look at what was changing there. And without going into the details really of the analysis, I just want to sort of move straight into what we found. We found a number of genes related to cell morphogenesis, geo terms, that seem to be exhibiting different dynamics. So not necessarily differences in their absolute levels, but in the way and the timing that they were coming on. So we could see that these tended to be turned on earlier in gorilla than in human. And as I mentioned, the gorilla seems to be undergoing these shape changes earlier than in human. So these were the genes we were most interested in. And so to try to get at which of these might be really, sort of regulating this process, we then looked at genes in this group that also overlapped with known transcription factors, and we came out with a list of 8 transcription factors. We focused in on ZEB2, this one that I've highlighted, because ZEB2 is also mutated in a human condition where brain size is too small. So it's a condition called Mowat-Wilson syndrome. These patients have microcephaly. So that suggests this is a very strong candidate gene for being involved in this process. So then work that Stefano carried out before he left the lab, he looked at ZEB2 levels and just confirmed what we saw in the RNA-seq data, showing that it was coming on earlier in the ape than in the human. And then also looked at whether ZEB2 is being expressed at the right time in the right place. He found that it was present in these neuroepithelial cells that have not yet become radioglia, so they're not yet expressing GLAST. And also found that if he mutated ZEB2, he could see an effect on neuroepithelial to radioglial transition, namely the staining of occludin. So occludin is a marker of tight junctions, and what he found is that this, when ZEB2 was mutated, you see an increase, so a more extended occludin. And normally during this NE to radioglial transition, occludin should get very, very restricted only to the apical surface. We can see here it's maintained along the apical-basal axis here. And that also fits with what's known about what ZEB2 does. So ZEB2 is actually most well known as a master regulator of EMT, so epithelial to mesenchymal transition. And in fact, this transition from NE, from neuroepithelial to radioglia, mimics this transition it shows quite a number of features of EMT. So you see a downregulation of occludins, you see an upregulation of things like N-cadherin and vimentin. Vimentin is actually a marker that's often used for radioglia. And so ZEB2 seems to be regulating this process, we think. So how is it then doing that differently in human versus ape? This is work that Sylvia and Magda Sutcliffe in the lab did together. Basically, what we wanted to do was to try to just shift ZEB2 expression timing and see if we could change the phenotype of these organoids. So what we did is to try to essentially express ZEB2 earlier than it should normally come on in human. And so to do this, Magda developed this nice strategy for controlled induction of ZEB2 using a doxycycline driving Cree. And we can get very nice induction of ZEB2 when we put on doxycycline. And when we do this so that we shift ZEB2 earlier, we actually could immediately see, just even on the bright field, that these organoids look different and actually look a lot more like the ape counterpart. And we can see that the cell shape also seems to already be transitioning, just like it is in the non-human ape, whereas in the human it's still more columnar in shape. And again, I think the ZO1 really demonstrates this the best. You can see these apical contacts have restricted already in the human. So we've sort of apeized these human organoids just by playing around with ZEB2 levels and timing. And so this is sort of the summary of this work, showing that we've identified this transitioning neuroepithelial stage in humans and other apes that seems to be delayed in humans. And the transition to this stage involves ZEB2 and its regulation of tight junctions. And also, I have to say, there's also actin constriction going on here. And I didn't have a chance to get into that, but we also have data looking at that. And this leads to an increased number of progenitors, so basically an increase in the founder stem cell pool that then ultimately would lead to an increase in the number of neurons. And what this would predict is that this would affect neurons broadly across all the different cortical layer types, so that what you would have is a tangential expansion over the surface of the brain, while the gray matter would still be the same thickness. And that's exactly what you see in adult human and ape brains. So with that, I want to thank the people that did the work. So I think I highlighted all the key players throughout my talk, but I've also circled their beautiful faces here. It's a really fantastic team to work with. I also want to thank my collaborators, of course, and my funding, and I'll be happy to take questions. Thank you.
So thank you very much, Madeline. Okay, you already stopped sharing your screen. So I will, very beautiful talk. I will start immediately with a question because you have some slight time restriction. So the first question is from Fernando Alcina, and he's saying, very nice talk. Maybe I missed this, but have you tried different male-female cell lines to discard that the differences that you have found are not partially due to the intrinsic cell line properties beside the sex?
Yeah, so I think it could be worth looking at more cell lines. I have to say, I think, of course, there are cell line differences, absolutely. But actually, the fact that we don't see differences between the male and female cell lines in the features we're looking at. So I should stress that, I mean, we've looked into this a lot. We've done a lot on different cell line differences and stuff. And what we see is that the biggest differences between cell lines tends to be at the earlier stages. So in terms of like just neural differentiation and whether they make neural tissues, and then what brain regions they make. But once they make a particular brain region, and you can be sure that you're looking at that brain region, so using a number of different markers, we have sort of a standard set of QC steps we go through with these organoids. Once we know that we have cortical tissue there, then actually things like cortical thickness, TVR2 numbers, different cell type numbers are quite reliable across cell lines. And this also kind of is mimicked in a recent paper from Barbara Treutlein and Gray Camp, where they also showed that when they look across a number of different cell types, donors, they find that the biggest variation, they do this by single cell RNA-seq, is in terms of identity. But once they make that identity, so if you look at just the 4 brain cell types that are made across different cell lines, then they overlap very, very nicely. So the male, female, and then so basically the differences that we are seeing here, we don't seem to see differences between the male and female cell lines. And actually, we see the same effect using these hormones on the male and female cell lines.
Okay, thank you. Then the next question is actually from Zoltan Molnar. Are there studies on brain size or on the prevalence of autism in populations of androgen insensitivity syndrome cases?
So androgen insensitivity, so I don't know about that, but what I can talk about is actually polycystic ovary syndrome, yeah, PCOS, where actually, so these are women who have high levels of androgen. So they're producing, for whatever reason, higher levels of androgen than normal. And babies born of women with PCOS have a higher risk of autism. And so I think that's quite interesting and kind of fits with the findings that we're showing. And so the other thing that I think would be interesting to look at is maybe the effects of androgenic environmental, compounds that women might be exposed to. And then the other thing I'd like to mention as well is actually VPA. So valproic acid is a drug that some people take for, for epilepsy, for example. And it causes quite severe neural tube defects if taken very early in pregnancy. But there's also evidence that even if taken later in pregnancy, that it leads to an increased risk of autism. So that it does, I think we are on to something here, basically.
Okay, thank you. Then a question from Bassem Hassan. Actually, two questions. He take the chance to get a second question in his first one. So the first one is very nice talk. Are the neural cells of organoids resistant to SARS infection regardless of organoid age? I'm particularly curious about younger organoids. This is the first one. And the second one, I will ask you later.
Okay. So so that's a very good question. So we tried on a range of ages. And actually, we were thinking maybe they're not old enough. So we tried very, very young organoids, so talking like 30 or 40 days, which is quite young for our protocol. But we also tried on our air-liquid interface cultures, which are, so I'd have to check exactly how old they were, but I think they were, on the order of 100 days or so. But actually, that's not even really the oldest we've gone. So I think it'd be interesting to try on really, really old ones, like year-old ones. And so maybe it's a question of that. Because one thing that's very interesting about the choroid plexus is it seems like the choroid plexus organoids can mature maybe faster than the rest of the tissues that we're generating in these organoids. And we can actually get cells that are really mature, I mean, that look like pediatric or even adult choroid plexus cells. And so that's also a very interesting question. Could it be that the choroid plexus is so susceptible in these organoids because it's able to mature so much? We have to look at that.
Is that the same when you have basically the choroid plexus spontaneously forming in the normal cerebral organoid? So the speed?
I don't know. Actually, don't know. Yeah, so that would be an interesting thing to look at.
Yeah. So his second question is a technical one and is about the evolutionary differences. It is clear that the culture media use, concentration, and all the inducers are not optimized for human neuroepithelial cells and are just slightly off for 8 neuroepithelial cells. So if the media are optimized for one or the other one.
So I can tell you that we use the same media even for mouse. And actually the components in the media are mouse derived. So they're actually equally distant from human and ape. And so I think it's a good thing that we're actually not using any human components in there. The only difference that we have to make to the protocol, and this is true for especially when comparing mouse and human, like when you're comparing mouse and human, is the timing. And the one difference and so the way we grow our organoids, this is something I probably should stress more in my talks actually, is also we adjust timing according to the cells. So we don't just blindly follow protocols without looking. So we always look at the organoids and adjust it accordingly. So you'll notice in a lot of our protocols as well, we'll say, move to neural induction on day 4 or on day, depending on the protocol you're looking at, but on day, let's say 5 or 6, We don't say just day 5 because it depends on the morphology. And so likewise, we do that across the species. And what we find is, for example, with mouse, we have to drastically speed everything up, but we can use the same media. Now for human and gorilla, we could use the same exact timing. The one thing we had to change slightly was for chimp. So for the chimpanzee, we had to move neural induction a little bit earlier. So the embryonic body stage is slightly shorter for chimp. And why that is, it just seems that they progress through that embryonic body stage a little bit faster than the human and gorilla. It could be a cell line thing, but I think it's very interesting because actually gestation is different. So the chimp gestation is 1 month shorter than human, and gorilla is sort of in between human and chimp. So that could be an explanation, but we definitely need more data.
So another question from Isabel Caillet, I'm not sure I pronounced correctly. Has this increase in excitatory neurons been observed in real human brains? And would this be human-specific or a general effect that should be also observed in mice?
So in order to see it in real human brains. So the other thing I want to stress is also that by putting on androgens, we are forcing, we're forcing we're forcing androgens, whereas you don't know what a given male embryo was exposed to during in utero development, right? So I think also the fact that this seems to be through hormones also fits very well with the fact that males and females are highly overlapping, because you can see how with a hormone, something that where you can have different levels and different receptiveness of the cells and stuff, you can see how it can be kind of titrated, and you can end up with a spectrum. What that also means, though, is that in the normal population, there is going to be quite a spectrum. And so if you take a given male brain and you look at excitatory neurons, you don't know how much androgens it was exposed to, and it may be at the lower end. So because of this huge overlap, you would need a very large number of brains to be able to see the population level effect. And so, and to count excitatory neurons, you can't do that like on an MRI or something. So it'd have to be postmortem analyses. And access to that number of postmortem brains is just not doable. So until something on that front changes, I think we won't know. What was the other question? Oh, if it's human specific. Yeah, so the interesting thing that is there, so I mentioned that DHT can't be converted to estrogen, and the reason why I mentioned that is because in mice, this has been studied much more extensively in mice, and in mice, it's actually estrogen that masculinizes the brain, so during development. So what happens is the embryonic testes produce testosterone, and then testosterone is converted to estrogen by an enzyme that's present within the brain. But in humans, that enzyme is called aromatase, doesn't really seem to be expressed. And in fact, we don't see it in our organoids. And the fact that DHT shows the same effect as testosterone in our organoids suggests that it's not through estrogen, because it can't be converted to estrogen. And also the fact that estrogen doesn't show the same phenotype as testosterone suggests it's not through estrogen. So yes, what I'm trying to say is there does seem to be something human or maybe primate specific going on here.
But maybe a similar mechanism without using the same cascade. It could be also in mice, but then studies should be done in litters that are completely female litters and male litters, because otherwise the hormones could maybe go through between embryos.
Yeah.
So another question from Esther Serrano. Great talk. Did you mention whether female and male organoids respond differently to testosterone? I think you mentioned that.
Yeah, so we didn't.
Is there some intrinsic property in neurons that make them respond differently?
Yeah, so we didn't see a difference between the male and female cell lines in their response to androgens. So that, I think, is also, again, going back to this idea that it's highly overlapping between males and females, and you could see how different levels of these hormones could affect male and female embryos equally.
So one last question from Sylvia. Could the smaller size you observe for the non-human primate organoids be due to the quality of the starting iPS cells or the protocol used, that it is maybe better for generation of human organoids? I think
that's related. I mean, of course, we checked the cell lines. We also have multiple. I focused in on, but we for the quantifications and such that we did, we quantified across different cell lines. So we have multiple gorilla, chimp, human, and the phenotypes we're looking at are really where you can see, the human cell lines are bigger, the chimp and gorilla cell lines are smaller. It's reliable and at the species level.
And maybe I have also one short technical question. So giving hormones to a petri dish is different than the physiological concentration that you would have in during development. So how did you define the concentration?
Yeah, so Eva, so I have to ask Eva exactly what the concentrations were there, but she and it's in the bioarchive. I think it's detailed more in the methods there. But because we know there's this fetal surge and we know what the amount is of that, the circulating hormones there, that's what we tried to mimic. We also compared with existing studies that have been done. Mm-hmm. And I think there's also been more work in terms of how much you need to give in a tissue culture vessel, in previous studies, in order for the cells to experience a particular concentration. Because actually, the other thing, these hormones are they tend to get absorbed by the plastic a lot. And so I think we had to give a little bit more, but we're in the physiologic range. Now, DHT is more potent, so we also actually decreased its amount by 1/3, I believe.
And did you try to do the same in 2D, so to see if if it's necessary actually to have a dimensional structure, or
That's a really good point, actually. So I think there is work being done by others in 2D cultures, but they're focusing more on neuronal, neurons themselves, neuronal maturation and morphology. And so, yeah, I think it would be interesting to look in 2D. Maybe it's not necessary to have organoids for this. But the nice thing about organoids is their organization, especially in terms of the excitatory and inhibitory neurons. So I think from that perspective, it gives us a sense of that kind of balance.
Good, so there are no more questions. So thank you very much for your wonderful talk, and also for answering all our questions. And yes, have a good evening to everyone.
Thank you. Thank you very much. Bye.
Ciao. Okay. I'm in bottom left. And broadcast, yes.